Thanks. So I'm Josh Morgan. I'm an assistant professor at Washington University in St. Louis. I'm a neuroscientist. I have a neuroscience lab. I've been doing, image analysis for a long time and working with array tomography. And we do a lot of large-scale electron microscopy in the lab. So what I'm going to be talking to you about today are some of the image processing steps that makes array tomography, at least for large-scale EM, work well. And what I'm kind of imagining as the audience is people who have some experience with EM and this kind of imaging, but are wondering about scaling it up from 2D to 3D or large 3D volumes. Okay, so there's a lot of image processing that can go into this process and really, making the data usable. So here's about 14 steps. I'm sure I missed some from automated acquisition all the way to data analysis. And I'm going to try to go through actually most of these, which gives me about a minute and a half per step. So I'll try to be efficient, but not totally confusing. I've highlighted these two in red though. And that's because these are the two steps where the image processing can really become a bottleneck or a barrier to getting the biology. So a lot of this stuff is useful in automating the process and making the process better. But image alignment, as Mark mentioned, is really critical for array tomography and can be a real challenge, and especially as you scale up and acquire larger volumes, both image alignment and automated annotation can be the most time-consuming steps of this whole process. Okay, so there's pretty wide variety of ways of going about tackling all of these different steps. So one approach is to write your own code. So we do a ton of MATLAB and now more Python code to deal with some of these things. It's good for some labs, not for others. There's running someone else's code, which is worth figuring out if you can get a little bit of Python expertise. A huge amount of useful code is available in GitHub. The standard now, even if I'm publishing a paper that's not a techniques paper, that's just a biology paper, we put the code up in GitHub and make it available to people. So that's an amazing resource that's getting better. There's free software. So ImageJ is great. The Fiji package of ImageJ has a lot of really useful tools for array tomography and large-scale EM. There's other tools out there. I'm really hopeful for Napari as a hub, mostly because it's Python-based as opposed to ImageJ, which people have to learn how to interact with the Java, which is, not the average graduate student's expertise. So, again, these tools are out there, and especially with things like Napari becoming a hub for other people's code, there's a lot of really great free stuff available. There's commercial software. There's, free and paid versions of WebGnosis you can use. And then there's finally image processing contractors. So Scalable Minds, Zeta, Google, Ariadne, these are groups of experts that you can collaborate with or pay to do the analysis for you. So, which of these approaches you take depends on your expertise, how much time you have, and really pretty critically on how large your data sets are. So for my lab, that threshold is about at a terabyte. So when we have a data set that's more than a terabyte, we send it out to another group like Scalable Minds and have them do the alignment. Because at that point, it's no longer a question of just, do you know how to align two images? It's, do you have a data manager who can keep track of all these files and keep track of all the alignments and keep the server running and keep the cluster running and do all of these things, which for me at least, it's better left to the experts. So if you're running a center, it makes sense to have an expert data manager. If you're running a small lab and you can do most of this stuff yourself, there might still be some steps where it's worth just contracting out for it. Okay, so now I'm gonna try to go through some of the steps for image processing. The first step is to take images that are amenable to image processing. And so we'd all like low noise, high resolution images that AI would love to see and figure out and give us good alignments and segmentations of. we have to do some trade-offs there, but one of the things we can do for free is just not to saturate our images. So, this is a nice contrasty image for displaying on a PowerPoint presentation, but I would not want to acquire an image like this on the electron microscope as it goes into the pipeline. So these are the saturated pixels. The image is inverted, but basically the blue are all the zeros, the red are all the 495s. So this is a 12-bit image. So all these blue and red areas, we've lost information. And we haven't just lost information, we've also basically applied a nonlinear filter. So if the signal is going up in this steady black line here, we're encoding it as this dotted red line here, and we're really only encoding part of it. And that means we've made some arbitrary decisions that image processing code downstream will have to deal with. And it's kind of, again, it's never good to throw away that information, but particularly if it's going into a pipeline where you're doing lots of automated analysis, it's nasty to have these weird edges there. So when I'm acquiring an image, I probably want it to look more like this. I'm making sure the bounds are far enough apart that I can encode the whole thing as close to linearly as possible. It's also, if you've mostly done 2D stuff and now you're going to automating a 3D array tomography pipeline, it's good to be even more conservative in this direction because you don't know how much contrast is going to change from section to section where various imaging conditions might change. So being conservative and really making sure that you aren't going to end up with thresholded images or saturated pixels at the end is pretty helpful and important. Okay, so the actual image acquisition process, if you want to automate it, involves a lot of image analysis steps. So I'm going to talk to you about a pipeline that I developed with Ken Hayworth in Jeff Lickman's lab. So he wrote a program called Wafer Mapper. So we're starting with wafers that have about 200 sections on them here. So these are 4-inch wafers that we pop into the microscope. So the first step is just finding all of those sections. So these are sections labeled by little red crosses here. You then let the microscope drive to each of these sections and acquire an overview image. So this is a low resolution image of the whole section. And before we start on the high resolution imaging, we align all of these low resolution images so we have a 3D map of the data set. So that does a couple things for us. One, it lets us make smart decisions about what parts of the tissue we're going to image at high resolution. It also lets us know if we did some optical imaging beforehand, we can line up the optical images to this low resolution EM map and direct the imaging in that way. We can choose a region of interest from this 3D map, define some mosaic like this. So each of these red squares is a tile that we're going to acquire. We can also, when we tell the microscope to drive to that position, it can take a picture and say, is this position I'm at right now, does that really look like where I'm supposed to be in this 3D map? And it can do some extra adjustments there. As it acquires these tiles, it can also look at those tiles and judge the quality of those tiles. So for each of those mosaics, we get an image that looks like this. This isn't the final product. This is kind of a on-the-fly pre-stitched image with these quality values written out on the corners here. Now the quality evaluation does a couple things. So first, if the quality value drops low, the microscope can retake the image automatically, which is always nice. But it also just allows us to monitor how the image run is going and look back on previous quality of imaging series. So, this wafer, we might do an imaging series that takes 24 or 48 hours. Before we pull the wafer out, we can do a quick run through this stack of pre-stitched images with the quality evaluations and make sure, did it really image the area it was supposed to image? Were there any problems? Was there drift in signal to noise or focus that we need to go back and retake some images or address some issue with the microscope? And so in general, I really advocate for having these automatic ways of evaluating image quality when you get to the point where you basically are producing more voxels than anyone can look at least while it's acquiring. So, this this method of image evaluation, it's a little kernel we wrote that compares structured contrast to unstructured contrast. You can use Fourier analysis, signal-to-noise analysis. You can also use deep learning to monitor this, which is nice because you can kind of not worry so much about what the microscope is doing and just from the biologist's point of view, decide what is a good image and what is a bad image and what does that look like to a convolutional neural network. So this is a network that we trained in the lab to evaluate images. In blue here is the user score. So you have bad images at 0.3, good images at 0.7, and in green is the prediction of the network. So in this case, it's doing a very nice job of recreating what the human user had assigned to the quality. And that's true for our test data set. You can look at the human scores and the predicted scores very close. So this gives an automated way, if you're doing an image series that might be tens of thousands of individual image tiles, you can say, you know, are we producing a lot of great images or crap or something in between, and look at what days things may have gone wrong and figure out why. Okay, so assuming you have your images and they're still not quite as nice as you'd like them to be, there's a lot of ways you can clean those up. These are kind of pretty standard image processing techniques. You know, a good one is always a median filter. So most of the noise in the SEM is uncorrelated noise where you want to throw out outliers. Median filtering is great for that. So you can take an image that looks like that and do some median filtering. You know, with all of this stuff, you want to be transparent about what you're doing to the data. And there's going to be a trade-off between resolution and signal to noise. But very often, a little bit of median filtering really helps us see things better. There's much more sophisticated versions, and people are having more and more luck using deep learning to clean up images. So the nice thing about training a deep learning network to improve your images is it's really easy to generate the training data. You basically take a good picture and take a bad picture, and those the same thing, and tell the network, figure out how to turn this bad picture into a good picture. So, you know, people are having a lot of success with that. It can be a little bit of a black box. There's another place where your images can get filtered, though. Sometimes the microscope is secretly filtering your data. So it's worth looking into the scope and figuring out if there's an option for turning off filters or even talking to the engineers. Because oftentimes it makes a lot of sense when you're building a detection system to build a low-pass filter into it, because they're assuming that, you know, the noise is this high-frequency noise, and your image dwell time is going to be relatively slow, and, you know, filtering out extreme values early on is going to be fine. If you start to push your system to doing very fast imaging, you know, for our system, it might be, you know, something like, you know, 100 nanosecond or 50 nanosecond dwell time. You start to enter a range where maybe the engineers weren't really expecting you to image that fast, and these filters can do very strange things to your data and your downstream analysis. Okay. Now, just generally, filtering with image frequencies can be useful. So this is a pretty common thing that you see when you're doing EM imaging, that you have vignetting, the corners are dark. If you're zoomed in, that's not such a big deal. When you're trying to acquire large fields of view, you start to see detector effects and beam geometry effects. And if you try to make montages of these, one, the montages look weird because you have these weird dark corners everywhere. But you're also creating artificial edges all over the place, which can really cause a lot of problems. So this vignetting is a very low frequency artifact. It's very different from most of the biology you might be interested in. And so it's easy to pull out in frequency space. Most noise is very high frequency artifact. Usually it's pretty different from the biology that you're interested in. So bandpass filtering can be a good way to get rid of these kind of extreme frequency artifacts on either edge. So, you know, in 2D space, this is what the bandpass filter might look like. Kind of a difference of Gaussians. You have some central frequency that you're interested for your signal and a larger frequency that you want to get rid of. And you can use FFT to do the bandpass filtering and very quickly pull out things like vignetting. Or another artifact we have is just kind of left to right difference in contrast due to stage conditions. Okay, so we've got nice looking images, good signal to noise. We put them together and we have this weird 3D thing where the brightness and contrast is changing between each section. So histogram correction. If your histogram of your image is nice and Gaussian, this can be pretty straightforward. You have a mean, you have a standard deviation, two numbers that describe this distribution for each image, and you can just go through each image and say, I want the means and standard deviation to be the same for each image, and you can get a nice matching and smooth that out. Most of the time, the distribution of values might look more like this. You might have areas of pure resin in your tissue that have very low values and areas of myelin that have very high values and some weird statistical distribution in between that varies from different regions. And just using a standard distribution may or may not work. So what we use for almost everything is basically sorting the values. So we'll first median filter the image, so to get rid of weird outliers, so signal to noise won't be an issue. Then we sort the values in the image and say we want 5% of the gray values to be above this point and 5% of the gray values to be below some point, and it stretches it out equally. And that works well for most of our data sets most of the time. Okay, so now we're going to get into the image alignment, which is the real kind of challenge for the array tomography. And there's a few different kinds of image alignment that are worth talking about. So first of all, there's stitching. That's just if you're creating a montage with tiles, you have to combine these tiles in some way to get them to line up and get rid of these borders. And then there's aligning section to section. So generally, for array tomography, stitching the tiles should be a really, really easy step. If you have your sections on a solid substrate and your beam is stable and things aren't charging, which for the most part, if you have on a conductive substrate, you should be okay, then the image you take of this little blood vessel from tile 1 should be pretty much identical to the image you take of this blood vessel for tile 2. So the matching problem is really simple because it's basically these two identical images that just need to be fit. And the actual transform you have to find is also pretty simple. It's usually just a little bit of wiggle in X or Y to make up for some stage variation on the nanometer scale. So this normally should be okay. If you're having problems with this, you probably want to look more at something being wrong with your microscope than a image processing issue. Aligning section to section is a different matter. So if you're collecting ultra-thin sections, first of all, there's compression. Now, you might have somewhere between 5 and 35% compression for individual sections, depending on the resin and how you're cutting it. And that compression can vary from section to section. So a really common artifact when you look at aligning these stacks is your images might be well aligned at the top, and then at the bottom it's jumping up and down because there's a different amount of wide compression, cutting compression that's not being compensated for. So you might also have wrinkles in your tissue, which can be really complicated and cause weird distortions. So all of those things make this potentially more difficult, and all of those problems get more challenging the larger the data sets get. So I'm going to spend a little bit of time just walking through some of the terminology for image alignment so we can talk about what is the best strategy for different kinds of data sets. So we're going to start just going over what is a linear transformation. Okay. So here we have our untransformed image. We have reflection, rotation, translation, uniform scaling, non-uniform scaling, and shear. All of these are linear transformations. It means that lines that started out parallel in the first image are still going to be parallel when you transform them. The nice thing about linear transformations is all of these transformations can be represented in this nice little 3x3 kernel where reflection, rotation, scaling, and shear are encoded. Did I say something wrong? No.
Yeah. She's enjoying a lot.
Okay.
You're good. Carry on.
So reflection, rotation, scaling, and shear are encoded in this square here. Translation is encoded over here. And with simple matrix multiplication, you can turn all of the XY positions in this image into any combination of these transformations here. So it's really nice, elegant, if that works out for you. There's kind of two versions of this to think about when you're looking at software. So a rigid transformation is just the rotation and translation. An affine transformation includes all of these combinations. And you would think, well, why don't we just always use an affine transformation if it's easy to code in here and it can do all these great things? And that's because the more constrained your alignment is, the better. If all you need is rotation and translation, then you only want to solve for finding the rotation and translation. And that's particularly true as you get to larger data sets. I don't know if this is true for other people, but definitely my initial experience of trying to align large image stacks is I end up with something that looks like this in the corner. And I'm pretty sure that's not the right solution, but it just accumulates error and turns into this squished little diamond. So anytime you can constrain this and say, well, I only want to look at plus or minus 5 degrees of rotation, or I want to look at non-uniform scaling, but only in this dimension, you're going to have a quicker and more reliable solution. So for our data sets, these different sources of transformations have different causes. So anytime you're doing automated imaging of your array, you have to deal with rotation and translation, that there might be some slight error in where the section is laying relative to the microscope. This non-uniform scaling is important for the reason I said with compression, that even under the best conditions, you might have this kind of squeezing as you do the cutting that can vary. If you can solve these two issues separately, you're more likely to have a robust solution. So we might do something like first finding the rotation of the section, and we can use the section overview or kind of a preliminary alignment to find that rotation. Then fix the compression in one dimension, just figure out what Y factor fixes the compression for that section, and then do an additional rigid transformation. So the final rigid transformation, So the final rigid transformation will hopefully get you to where you want to go. Now, how do you actually decide what that transformation should be? There's three basic approaches. So the first one is correspondence points. If you only have a couple sections, you can say, this point in this section definitely matches up with this point in this section. This point definitely matches up with this, and you just have, 3 or 4 pairs of correspondence points and solve for that, and you get your alignment. Usually you do this manually, but you can also have things embedded in your tissue, for instance, that are easy to identify that can help you do this solution. Next method is cross-correlation. So, for instance, you could rotate the images, calculate the cross-correlation. The peak of the cross-correlation is your solution for the best XY transformation for that rotation. This works really well if your images are really similar. So if you have small sections without much distortion and the biology is not changing very much, oftentimes cross-correlation works pretty well. The more the sections change and the more distortion there is, the more this tends to fail. And so the approach that's probably more robust and definitely our starting point is to use features within the section. So this is matching unpaired features. So These features can be mitochondria or membranes or just little contrasty dots. But if you find a bunch of those in section 1, a bunch of those in section 2, there's some ways to solve for matching up these two groups. And the nice thing about this is that you're not necessarily finding the best average transformation to get these red dots to match the blue dots, but you're trying to find some transformation where a subset of these are really, really well paired. And that's tends to be the most robust solution for our kinds of data, that there's some things that are constant from section to section, and then a whole bunch of stuff that you're going to want to ignore. And this matching of unpaired features is a good way to go about that. So I know this is kind of a vocab lesson, but for the people who haven't done this before, the reason is the next thing I'll recommend is if you have your first EM stack that you need to align, you can go into Fiji, ImageJ, open up Fiji, go to plugins, go to registration, and then go to linear stack alignment with SIFT. So now we all know linear stack alignment with SIFT means you're doing this kind of linear transformation here, and you're using these SIFT points to find which part of this image match up with this image. If you're doing a small volume in a small area and you don't have much distortion and you're lucky, this will solve your alignment problems. Things get much trickier when you get larger areas and there's distortion in your sections, and wrinkles are probably the most horrible problem because you have really sudden changes in the transformation, and then you have to do some kind of nonlinear transformation. And this gets harder. Both the transformation is more difficult, and the larger the volume is, this can explode in terms of computational requirements. So we're talking about elastic registration or warping or nonlinear or mesh. Basically what you're doing is you're finding your linear transformation for lots of different points in your section, finding the best transformation for this point, the best transformation for this point, and then finding a clever way to solve this mesh of how you transition from this alignment to this alignment to this alignment across 2 dimensions. And that can be really computationally intensive to get that right. And you can do that yourself. Again, so Stefan Soffield actually created a ton of great tools in ImageJ under elastic alignment. So again, you can go to registration, go to elastic, and elastic stack alignment. Those tools exist, and you can tune the parameters, and you can get that to work for you, and you can do these things in TrackEM 2, which is also part of this plugin. It's just that as your data gets larger and larger, this becomes a more difficult problem where you might need someone else's expertise. Okay, so one more kind of alignment, that if you want to align your data set to another data set of the same piece of tissue. So our strategy in the lab is, even if we start out with something like 2-photon imaging, after we do aldehyde fixation, we then create a dense feature optical map of the tissue. So in this case, we have DAPI staining of a piece of brain, we have reflected light showing us myelin, we have synaptic boutons labeled with CTV. This kind of image matches up really well with a low resolution image of the EM. So this is the same piece of brain tissue section, a few hundred microns across, and we can find the fibers, we can find the blood vessels. We can zoom in and match the blood vessels at higher resolution. We can zoom in again and match the cell nuclei. And these chromatin clusters are really great for matching between cells and identifying cells and get down to the micron scale. So, this doesn't require any particular software on its own. Most of this can be done manually, but as you get down to finer and finer scales, you might need some kind of assistance. So we had in one of our last projects kind of a semi-automated pipeline where we would find the transformation, tweak the transformation, and find more correspondence points. So this is an example where the EM reconstructions of dendritic arbors are in gray here. And all of these points here are places where we found the same neurite in the optical image. In this case, 2-photon calcium imaging. We would find a correspondence point that would refine the image, find a new correspondence point that would refine the alignment more and more. There's tools available for this. So, Big Warp is something people use a lot for this kind of 3D registration of data sets. Just in general, this Bioimage Informatics Index is a really great resource for looking for this kind of software, aside from just going to someone's GitHub page. There's multiple tools for finding these things. Okay, so I have about 9 minutes left, and so I'm going to try to get into one of the more complicated areas, which is image annotation and segmentation. So the simple version of this is doing manual tracing. So you have a digital coloring book. We really like the program VAST that Daniel Berger wrote, where you're basically going section to section and you're painting features and putting those together in 3D for rendering. My lab still does this a lot. Would rather not do this. This can be a very time-consuming process, but it is at least robust. Another approach is heuristic. So this is more traditional automated image processing. So this is another piece of mouse brain here, and I've first filtered for size. So in this middle image, we're looking for blood vessels. So I found the bright spots of a certain size, and then thresholded it, then first frequency, then threshold it, then do a size filter and a shape filter. So this kind of combination of frequency, threshold, size, and shape can give you the blood vessels or cell bodies. It's relatively fast. You don't have to spend a lot of time training a network. Almost anything that this heuristic method can do, a deep learning network can do. The nice thing about the heuristic approach is you actually know what it's doing, and maybe you can implement it a bit faster. But of course, what is really becoming more and more and more powerful very quickly is using deep learning to solve these problems. So this is a implementation that a rotation student in my lab did using Google's open source code base. And so this was actually in 2018, just segmenting all of the neurites, neural processes in a piece of EM data. And to get this to work, we had to take Google's network and do a little bit of extra training. Getting to this first stage was pretty easy. This was 7 years ago, 6 years ago, something like that. And we still have not applied this to a full data set. And that's unfortunate. And that's where we were for the past few years, where getting this code to work, or this kind of code to work on a small piece of data, can be pretty straightforward. Implementing it on a cluster for a large piece of data can be really, really challenging. And I think the software tools are getting better and better for making that work for small labs, but it can still be really tough organizing all of this code and organizing the data. So again, it has the same role that at some point, just the sheer size of the data, you probably need a professional or specialist to manage that. But we still use these networks for all sorts of things. So this is just some recent work by Massabank Tavakoli, the same person who did the quality check stuff, where we trained a network to find not just mitochondria, but mitochondria of the boutons of retinal ganglion cells. So it's not just using the morphology of the mitochondria, it's also saying this mitochondria is next to some large vesicles, probably that's what it's saying, and using some other cellular features to pull out a particular subtype of mitochondria from lots of different mitochondria. And this, was not a horrendous thing to train. Like, you have to get training data sets and collect that information, but it probably took a week of segmenting or picking out images of RGC mitochondria to create a training set that this network could then use to identify those mitochondria versus other things. So I could talk about some general deep learning stuff and the fact that deep learning can write its own code, but I think I'll skip to the end and then answer questions if anybody has any. Again, I would like to point out the work of Masa, who did a lot of the deep learning stuff, partly because she's now looking for her next position. If anyone wants someone with some experience doing this stuff as a staff scientist, let me know. So thanks for your attention. Any questions?
Thank you, Josh. Any questions from the audience? I have a question, Josh. So when you use the professional to help you to do the analysis, do you just give them the raw data, or you actually do the alignment first and then ask them to do just one specific process like segmentation or things?
Yeah, so usually what we've done is have a folder with either the raw data or slightly pre-processed data. Like we might do a little bit of pre-processing, but not alignment. And they give back the alignment, and then we've done everything else. And so that was with Scalable Minds a couple times. It's worked out reasonably well.
And so they have a pretty fast turnaround when you contract them to do things?
Yeah, it's been okay. For the large data sets, it might take us a month or two to acquire the data set, and maybe a month or two to get the data set back, and then a couple of years to analyze it. So if they're doing the alignment, that's definitely no longer a bottleneck in the process. So.
Okay, all right. There's someone else had a question in the chat and ask any resources to get started on the heuristic method you mentioned.
Let's see. If you're coding it from scratch, or you could download our code. I think the bandpass filters are available in ImageJ if you go to Process and FFT. So playing around with bandpass filters is really easy there, and you can see what kind of features things enhance or don't enhance. You can probably do size filtering and shape filtering through that afterwards, although everything that we've done is just custom code. I hope that helps.
I also have a question about when you mentioned about you get poor quality of images, that when you were pre-screening, doing the scale. So what are the causes for getting poor quality images? From sample processing or image acquisition?
So the main problem that we had is stigmation drift. So these are images acquired on the Merlin, which the Merlin size scanning electron microscope is really nice because it has a lot of flexibility for different kinds of imaging, but it's not necessarily the most stable beam in the world or column in the world. And so from image to image or over time, there might be a little bit of stigmation drift, and you can automatically stigmate. Sometimes that fails. So that's why automatically detecting poor quality images is nice because you can just tell the microscope to refocus or restigmate, but there's always a little bit of patchiness that gets through that, in our experience at least.
So that means you would just get some tiles will have poor quality, but some of them are okay. It's not the whole.
Yeah, and usually we'll have some sections of kind of borderline quality, and you have to decide at what point you retake the image versus go ahead with it.